in this video we will just discuss some genetic questions which generally comes for the MSc entrance exams, or these questions and these concepts of these questions are quite frequent and important also. So let's start discussing about that. First question is, in sickle cell anemia, which of the following hemoglobin subunits is affected? So we know in sickle cell anemia is a disease where the hemoglobin protein undergoes a mutation. Hemoglobin HbA and HbS, that is the mutated form, they have this alpha and the beta subunit. But what happens in the normal HbA hemoglobin protein is the beta subunit, which has the glutamine amino acid, and glutamic amino acid, which is normally present, will undergo mutation to produce valine at position 6 instead of that glutamine, and that will result in a mutation which is HbS protein, and that results in the sickle shaped RBCs due to deoxygenation. So therefore we are asking to the answer which of the following hemoglobin subunit is affected. We know alpha and beta is present, but beta subunit is affected, so the beta B will be the answer. Next question, dosage compensation in humans is brought about by the. Dosage compensation is talking about when we are talking about two different sex, that is the female and the male. We want that in both the sex we have an equal level of the X-linked gene products, and to maintain that, females who have two X chromosome undergoes some compensation, and it results in inactivation of one of the X chromosome just to equalize the difference. Okay, so this different kind of organisms have different strategies of doing this, and in this question the options which are given are inactivity of one X chromosome in females, hyperactivity of single X chromosome in males, hypoactivity of both X chromosome in females, hyperactivity of autosomes in females. Another important thing about dosage compensation is it happens to one of the or two of the X chromosomes only. Okay, this is a modification in only the X chromosome. So here in the last option autosomes is written, and that's why automatically this this question is this option is not the one, so we'll just cross it. And the other three all involves X chromosome modification, which is true, and all of these modifications does exist. Our question focuses on humans, so we have to answer that. Let's see where they apply to. The other three options, the inactivity of X chromosome in females, inactivity of X chromosome, one X chromosome, this is done in humans. This is our answer. Okay, this is done in mammals, so humans is mammals. We will answer, that's our answer A. But the next two, that is hyperactivity of single X chromosome in males, this takes place in case of Drosophila flies, and hypoactivity of both the X chromosome in females takes place in hermaphrodite worms. So we saw that all these three options are correct, does happen, but in humans we have to say it's inactivity of one X chromosome. A is the option. Next question, which of the following is most likely outcome of a cross between two heterozygous tall plants? We have been said that it is a two heterozygous tall plants, and they are asking the ratio which will be outcome from crossing them. And these two heterozygous are tall, that means they can be double T or it can be double T small t, both are heterozygous. So again they are asking for the ratio of the cross between the two heterozygous tall plants. So we know that since they are tall heterozygous, that means it will be a single capital T and a small t. that will be heterozygous, and they will give a tall phenotype because tall is the dominant one. But of course they are asking the ratio, and they have given us some numbers, so we have to tell them. Option number C, that is 24 tall, 49 medium and 25 short, is not the one, because if you see the Mendelian laws, according to Mendel laws, the two heterozygous tall plants, when they are crossed, it will be a 3:1 ratio, the genotype. Yeah, so obviously number C has 3 ratios, and that won't be the answer. So we will cut it first. Second, we will just observe the other 3 options. 63 tall is to 59 short. It's a ratio which is, they are very close. 63 and 59. So it definitely won't be a 3:1 gap as a ratio that we can say just by seeing it. Next is the last one, 53 is to 147. 147 is way more than 53, and again it won't be falling under 3:1 ratio. But if we see the 76 tall is to 23 short, that is, I guess, a perfect 3:1 ratio that we can check by adding 76 and 23, we'll get 99, and then when we 3/4 of 99 will be 76 and 1/4 will be 23, and that is how we can confirm that 76 tall is to 23 short will be the correct answer. Next question. In a cross between 2 black Labrador retrievers, the phenotypic ratio of the offspring is 9 black puppies to 3 chocolate puppies to 4 yellow puppies. And this is an example of the options are partial recessiveness, B incomplete penetrance, C incomplete dominance, and D epistasis. Well, the answer will be epistasis, and since incomplete dominance is talking about a 1:2:1 ratio, and here in case of this question given is 9:3:4, which we will see in none of them partial recessiveness and incomplete penetrance, but in epistasis. Right, the answer will be D. Next question, this is a good and easy question. 1 mapping rate of 1 centimorgan corresponds to recombination frequency of 1%. This is a fact. Yep. So A is the correct answer. Next question. Eye color of Drosophila melanogaster fruit flies is sex-linked, with red-eyed color being the dominant to the white eye color. And in a cross between a red-eyed male and a heterozygous female, all of the following progenies are expected, but except which one? Okay, we will do that and see. Okay, so they are asking a red-eyed male. So if we take the gene R, red-eyed, make it R, so we will write XR while here. And then since it is true that eye color gene is present in the female Drosophilus, so and a heterozygous female is present which has the genotype like X capital R and X small r. So we'll just cross it and we'll get XR X capital R X capital R, which will be a red-eyed female. So red-eyed fertile female, and that is A. We'll cross and see first is XR, that will be a red-eyed female and red-eyed fertile female, so that is option number A we can see. Next in this square we'll get an X capital R and Y, which will be red-eyed fertile male, which is option number B. Next third cross is, not third square, is X capital R and X small r. So this is heterozygous female, but since it has both the alleles, so X capital R is present and it is red is dominant to white, so we'll get a red-eyed female here only. And next another one is X small r allele and Y, so here it's a male, which will be white-eyed, because it's the small R allele is present, so it will be a white-eyed fertile male. So that's option number C. So the option which is not present is white-eyed fertile female, which cannot be present, which cannot happen. So our answer is number D. Next question. The pedigree below represents the inheritance of an autosomal recessive trait. What is the probability that individual 6 is a heterozygote? The question says that it is an autosomal recessive trait, the black one below in 7. So that autosomal recessive trait will be shown here as small a, if we consider it as an A allele. Small a. So when do we get a small a? Is when our parents are heterozygous. That is a capital A, small a. Both the parents are heterozygous. So if we do that cross here, capital A, small a, and here capital A, small a, then we'll get 4 squares and we'll get capital A, which is homozygous, capital A, small a, which is heterozygous, another capital A, small a, heterozygous, and a small a. So this small a one has been received by the 7, individual 7. So 6 can receive only heterozygote. What is the probability of 6 getting a heterozygote? Since there are only 4 options, 1 option is taken, and here two possibilities are there of getting. The possibility is 2/4 for individual 6 to be heterozygote. So that basically means 1/2. 2/4 is a half. So possibility of individual 6 being a heterozygote is half. That is option number B. The next question, the allele small l in Drosophila is recessive. Sex-linked and lethal when homozygous or hemizygous. If a female of the genotype capital L small l is crossed with a male, what is the ratio of female is to male in the progeny? So we'll consider a female heterozygous. Of course they have given the allele capital and small l, and we'll cross it with male. Okay, the male won't have a small allele, but the male X chromosome will have a capital L, since that is not lethal. So we'll take that and we'll cross, and we'll see that we have first got X capital L, X capital L. Then we will get an X capital L and X small l. Then we'll get X capital L and Y, and X small l and Y. So here we can see the small X small l Y will not survive, because that small l allele will be recessive and it will express itself in the absence of the capital L allele, and since it's lethal, will be dying. And the other, so in the result get 2 females who have that X capital L allele, and it's dominant, so they will survive, and the male one will survive, that is X capital L Y, since that L is fine. So we'll get 2 female and 1 male, and that is option number B over here. So answer is number B. Next question, a child with Edwards syndrome 18 trisomy, having his mother where non-disjunction of chromosome 18 occurred in the division of the secondary oocyte. What is the chance that a mature egg arising from this cell division will receive 2 numbers of chromosome 18? Okay, the options are 1/4, half, 1/8, and 3/4. So the mother cell which will give rise to the secondary oocytes is 2n, and undergoing meiosis 1, it forms the secondary oocytes, and then secondary oocytes gives rise to 4 such ovums, where secondary oocyte is a mature ovum, and on meiosis 2 it will give rise to 4 ovums, where 2 will be the polar bodies and the rest will be the mature eggs. So they have asked that, they are saying in the division of the secondary oocyte, where the secondary oocyte is forming, there is a non-disjunction of chromosome number 18. So if the mother was 2N, so it had 2 18 chromosomes. Now it will get in meiosis 1, it will give one of them to first egg and another one to second egg over here. But since non-disjunction has happened, one of the eggs will receive both the chromosomes and the other will remain empty without chromosome number 18. So this is in meiosis 1, and in next is meiosis 2. It will give rise to this particular egg. Oocyte will give rise to 2 such eggs, mature eggs, and they will have, since it has mitosis, it has replicated more, it will equally divide to both of the eggs, so both of them will receive the 218 chromosomes, right? So they will have, okay, and then the rest, the one which was empty, will not give any chromosome number 18 to any of the eggs. Next is one of this egg will now, it can go and fuse with the male chromosome, which will have only one of the 18 chromosomes, right? 18th chromosome. So now when these two will fuse, they will give rise to trisomy 18 child having 3 chromosomes of 18, chromosome number 18. Yeah, so here we can see that mature eggs are 2 out of the 4, and both of them has 18 chromosome twice. So out of 4, 2 eggs are having 2 numbers of chromosome 18. Therefore 2 out of 4 is giving us half. Therefore it will be answer number B. Answer is number B, which is half. So the next question is holocentric chromosome. The entire chromosome acts as a centromeric region, whereas if you see in case of the monocentric chromosomes, they have a single center in between the entire chromosome, which acts as the microtubule binding point, and that acts as a centromere, which bears the kinetochore, right? So in case of holocentric, they have the entire region. So our first option, that is chromosome with multiple centromeres, will be the correct option. So 88A. Next question. In crosses involving the different pairs of genes A and B, C and D, and E and F, so the following proportion of recombinants were obtained. That is, they are given as the percentage of recombination. A and B gene when crossed, it was 52% recombination. When C and D gene was crossed, it was 13.8% recombination. When E and F was crossed, it was showing for recombination frequency of 26.4%. Next, the pair of genes which are not linked is it A and B, is it C and D, is it E and F, or is it all of the above? A recombination frequency of 50% or more means that those 2 genes are unlinked and has maximum possibility to undergo recombination. So anything more than 50 or 50 will be unlinked. Here our option is A and B, which is showing 52% of recombination frequency, and the other C and D's is way less than 50, that is 13.8 and 26.4. That means they are linked genes. The unlinked genes are A and B. That is option number A is the correct one. Next question. Here, in a sample from a population, there were 65 individuals with the BB, that is capital B homozygous genotype. So next is 30 individuals with a capital B small b genotype heterozygous and 50 individuals with the small b genotype. The frequency of small b allele was. Here we are going to use the famous Hardy-Weinberg's equilibrium principle and assign that p square pq and q square their values. Here it is p square is 65 p allele. Then PQ, the heterozygous, is 30 and small b that is the Q square allele, B allele is 15. So in total that is 110. Okay. Now they are asking the frequency of B allele. Since we know it is here in PQ we will find B allele and in Q square we will find B allele. But in Q square we have to multiply it with 2. Yeah, because it is 2 times in a sample. The same is present in 2 chromosomes. So we will just multiply it with 2. Same with 110 into 2. Okay, that's 15 into 2 plus 30. In PQ it is found only once, right? Then divided by 220. That is 110 into 2 because all of them are found twice. So we'll get 30 plus 30 divided by 220. 90 by 220 gives us 0.27. So our answer will come as A, 0.27. Next question. Color blindness in human beings is an X-linked trait. A colorblind man has a 45X daughter who is also colorblind. The non-disjunction that led to the 45X child occurs in which parent and in which meiotic division is the question. The X-linked trait has the color blindness mutation. So, when a colorblind man has a daughter, that means that X is has the mutational small c. So when that male is crossing with a female which has heterozygous to color blindness, that time it will they will have a daughter who is colorblind. So the non-disjunction which will lead to the parent who had it will be mother, and in which meiotic division it is always the meiosis 1 where the actual reductional division takes place. So it will be the first meiotic division. Okay, so next question. The answer is C here. The next question. Considering the cross capital A small a, capital B, capital C small c, here we'll see the entire thing at the cross written for 5 genes with all the genes independent and they are sorting. The proportion of progeny will phenotypically resemble the first parent. Okay, so they are saying these are the 2 crosses with the genotype is given. Now the phenotype of the progeny will be similar to the first parent. So we will see what is the proportion. So here it's we'll do this for capital A small a and small a, since it's heterozygous, so that's why heterozygous into homozygous will give us all heterozygous. So we'll do for each of them over here. That is capital A small a and small a will give us 2 heterozygous and 2 homozygous. So we need a heterozygous because first parent is a heterozygous. So it will be 2 out of 4. For second one also we will see a homozygous into heterozygous. Again here 2 will be homozygous and heterozygous, 2 heterozygous. So it will be, for getting a homozygous we will get 2 out of 4. Same in case of C also, one is heterozygous, the other one is homozygous. Doesn't matter who is the one, but if we want to get a heterozygous since the parent is heterozygous, so it will be again 2 by 4. For 5 of the alleles we see the same thing repeating. We will so get 2 by 4 into 2 by 4 into 2 by 4 into 2 by 4, 2 by 4. Yeah, so 5 times will multiply 2 by 4, which will be half to the power 5. That is 1 by 32. Yeah. So our answer is option number A. Next is, what will be the probability of obtaining a plant with capital A, small a, heterozygous for A gene, homozygous for B gene, and heterozygous for C gene genotypes from trihybrid parents? Okay, we know that a trihybrid has 64 combinations and 8 possible gametes. So whatever out, it will be all out of 64. But how to calculate how many? So treating them as we will treat each of the genes as monohybrid, and we'll see that what is the probability of getting the plant with heterozygous capital A, small a from a trihybrid of this. So capital A, small a into capital A, small a will give you all of them. Here capital A, capital A, capital A, small a, capital A, small a, and small a, small a. So 2 of them will be heterozygous, 2 of them will be homozygous here also. So for the A gene we want heterozygous, so it will be 2 out of 4, right? So 2 out of 4 is half. Next is capital B, we want, but parent is having capital B, small b, capital B, small b. Again we will get 2 homozygous capital B's and 2 small b's. And then heterozygous capital B, small b, capital B, small b. From here also we'll get, since we want capital B, which is homozygous, that will be only 1 out of the 4, right? So in this case we will consider 1 out of 4. For the third gene, that is capital C, small c, we want from a capital C, small c only. So that will be again 2 by 4 out of the 4 parents, 4 gametes, which we will get. So again, 2 by 4 into 1 by 4 into 2 by 4, we will get 4 by 64. So 4 out of 64 is the answer. That is option number A. Next, corn has a diploid number of 20. How many chromosomes would be expected in A, that is a meiotic product, B, a polar nucleus, C, a mature embryo sac, and D, an endosperm cell? So 2n is 20. So here they are asking what is the ploidy level of meiotic product, which is n. So n is 10. We will get 10 as the first option. Next, for polar nucleus, it is again n, because polar nucleus is the last one, which is the immature one. So here also it's n. So again here it is 10. But a mature embryo sac has been fertilized with a 2n and n to give a 3n. So 3n will be 3 into 10, that is 30. And an endosperm cell is same, where one of them forms the mature embryo sac. So endosperm cell is also 3n. So we'll get again 3 into 10, 30 as the ploidy level. So option number A is the correct one, where it's 10, 10, 30 and 30. Okay. So this was our last question, and hopefully all the questions were helpful and has clear concepts, and how to attend these questions will be easier from next time. Thank you for watching.